Hello everyone, welcome back for part 2 of my lecture on the cell cycle. Where we last left off in the first video, again, was talking about the difference between chromosomes, chromatids, and sister chromatid pairs. I'll review this after I'm done the rest of the lecture today. So we're going to move on and actually talk about now what we refer to as the cell cycle. So the cell cycle, again, is what we call the period of growth and division of somatic cells. Remember, somatic cells means body cells, and they would, if it's a human somatic cell, it would be 46 chromosomes. So let's pretend you got a brand new little baby somatic cell that has just been born and created in your body. All right, it's going to grow over time, and eventually it's going to get to that point where it gets too big, its rate of diffusion decreases, and it's going to want to go through division. So that's really what the cell cycle refers to. Again, growth and division periods. So if we move forward here, a cell is going to have, as I just mentioned, two general periods that we need to understand what the difference is between the two. The first of the two periods is what we call interphase. Inter means between. So this overall is classified as the growth period of a somatic cell. After it's born, it's going to grow and do whatever its normal job function is. If it's a skin cell, it's going to help protect you from infections and viruses and ultraviolet light. If it's a heart cell, it's going to pump to keep your blood oxygenated. But they're going to be growing and doing whatever their main function is. Once it's done with this phase here and it has to go through division, if it can, it'll then enter division. All right, there's actually two smaller steps in division. We'll talk about that later on in another animation video. But for now, our focus is going to be on interphase followed by division, where that cell will divide in two and create two identical clone cells that are really referred to as daughter cells. But they'll be clones, 100% the same as each other. So let's imagine you are going to go through division. You would just start shaking and divide in two perfect copies of yourself. Those will be daughter cell versions of your original form. All right, so let's focus on interphase right now to finish out this part for the cell cycle. Interphase is going to be broken down into three smaller subphases that we're going to need to keep track of. All right, so for interphase, if you were to measure a cell's life as 100%, interphase is going to take up at least 2/3 of that somatic cell's life. So 2/3 is what, like 66.66%. All right, but it could even be as high as 90% of the cell's life is spent in interphase, because that's when it's actually doing its job and what it needs to do. So G1 will be the first official step in interphase. So you got a brand new baby cell which is created. G1 simply stands for growth. This is going to be a period of rapid cell growth, and during this time frame, the cell is going to be doing its job, as I keep saying, whatever its main function is. Now, as it grows, eventually it may get too big, where, as we said, that rate of diffusion starts to suffer. If that happens, it's going to enter what we call the S phase. S stands for synthesis. This phase will only begin if this cell passes something what we refer to as the restriction point, or the point of no return. All right, so the restriction point pretty much means the cell has now gotten too big. It will eventually die due to its rate of diffusion decreasing. So if it passes this point of being too big, its restriction point, it's going to want to officially start to synthesize its DNA in the S phase. So all of its DNA, or all of its chromosomes, are going to be copied. That's where I was saying earlier in the first video that the chromosomes will be copied. Each one will now look like an X, or a sister chromatid pair. So that's going to be all happening. DNA replication will happen during this S phase if the cell gets too big and passes this restriction point, or point of no return. The third and final subphase of interphase is going to be what we call G2. G stands for growth again, but besides a little bit more growth, I want to focus on what really new things are being done during this period. So the cell has gotten too big, past the restriction point, it's copied all its DNA. So now it needs to make sure that once it does go through division, the two brand new baby daughter cells will have everything they need besides the DNA. So that means all the organelles need to be copied. Multiple copies of the rough ER, the Golgi, the smooth ER, everything we discussed earlier this year, are all going to be copied so each brand new cell can get multiple versions of each organelle required. Also, you'll start to synthesize more and more proteins because these do everything in your body. They're enzymes, they're used for food, for building blocks, you name it. So the cell is going to make a lot more proteins. And finally, before it enters division, the last and probably the most important thing that's done is all the DNA that was copied during the S phase now needs to be proofread or checked for errors. If an error is found, a mutation, it'll be fixed before this cell can enter diffusion. Before it can enter division. If it cannot fix the error, the cell most times will just stop dividing and actually terminate itself. It'll actually dissolve itself by rupturing its lysosomes, which will release their enzymes and dissolve the whole cell. Now, sometimes a mutation or error actually may get by, and that's what leads to a mutation, which leads to disease or a multitude of other issues you might have. But as far as interphase goes, again, at least 2/3, 66% or more of a cell's life, break its life down in the G1 growth, S phase for synthesis. If it gets too big and passes this restriction point, it'll copy all its DNA. And then G2, the third and final subphase of interphase, will occur where now we're going to make all organelles. We're going to copy them multiple times. We're going to make a lot of protein that the cell can use for whatever it needs it for. But again, we're going to check all the DNA. So all 46 chromosomes that were copied, we're going to check it for errors and fix any errors if it can be fixed before we actually enter division. Now, a great visual is to look at a diagram like this. It doesn't matter where you start or what perspective. So right here at where my arrow is, let's pretend we're starting right here. Here's a brand new baby cell. All right, it's entering G1. I'll get to this G0 in a second, but it enters G1. And eventually, let's say right here at the tip of this blue arrow would be the restriction point. All right, that point of no return, where the cell has now gotten too big as it's been growing, doing whatever it needs to do for its own function. But it passes this restriction point, which means now it has to either divide or it has to die. So if it's going to divide, it's going to enter the S phase, where DNA replication will occur, we said, synthesize all the chromosomes. And then we'll enter the third and final phase of G2, which is where more additional preparation for mitosis, that is copying the organelles, proofreading, checking the DNA for errors, as well as making proteins. And then we'll enter the last third of a cell's life, a third or less of a cell's life, which is mitosis or division, which is broken down into prophase, metaphase, anaphase, and telophase. We'll talk about these in the next animation that I post. Now, you see this little G0 phase. We're going to finish out with this. Some cells, when they're born, they do grow, but they never keep growing to the point where they reach that restriction point right here, the point of no return. So they never get too big, and they never have to divide. So in essence, these cells could live forever. Or if they're never going to go through actual division, that means if you lose them, they're gone forever. So some examples of cells that once they're created enter this limbo of G0 and never pass the restriction point and never have to divide. Those would be things like brain cells, heart cells, or nerve cells. All right, any of those cells, once you stop growing, all right, usually your brain and your heart, at least, by the time you're about 12 years old, they don't grow anymore. They don't really get any bigger size-wise. So all those brain and heart cells will then enter G0, And at that point, you just have to try to maintain them. Live a healthy lifestyle, exercise, don't smoke, don't expose yourself to fumes that could cause brain cell death, etc. Nerve cells will, until you're done puberty officially and you're done growing, they can still elongate and grow. But that's what I mean by G0. These are cells that'll never pass the restriction point any longer. So they're never going to enter the S phase or G2. They're just going to stay in limbo. But if you injure them or they die, they're gone forever. So again, this is the entire cell cycle right here that we were just talking about. So to finish up, all right, if you have any questions, please email me immediately, all right, today or tomorrow, or whenever you get a chance. I'll get back to you as soon as I can. But just to finish up one last thing, I just want to go over those terms: chromosomes, sister chromatid pairs, and chromatids. So real fast, let's just pretend that here is the nucleus of a cell, and all the DNA exists as that spaghetti, as chromatin right now. All right, so you can't really discern any chromosomes in there, but they're in there. So by the time division, there are chromosomes in there though. So pretend that, I'll just start really easy. In that big mess of chromatin in there, there's one big thick chromosome. All right, and it would have a big, let's pick a different color here, I'll pick blue. It has a centromere in the middle right there. Now before division actually occurs, remember all of these chromosomes, 46 for us, would have already been duplicated. So now, if we could see them, they actually look like this. All right, they look like X's. I'm going to put my centromere right there in the middle too, right? So when I see a structure like this, I count my centromeres. Right now, I have one chromosome, but since each half of the X is an exact copy of that original chromosome, I have two chromatids, if I can try to write there properly. But as long as two chromatids are attached together, I'm going to call them a sister chromatid pair. So every X means I have one sister chromatid pair. So if I just did it one more time, let me draw another X real fast for you. Let me add my centromere. And if I were to look at these two X's right now, and I were to try to figure out how many of each of these three terms I have, I'm going to count my centromeres. I have 1, 2, so I would have 2 chromosomes. I have 2 X's, each has 2 sides, so I now have 2 sister chromatid pairs. And each X is comprised of 2 identical chromosome copies, or chromatids, so each half of the X is a chromatid. I have 2 X's, so I have 1, 2, 3, 4 chromatids. All right, being able to figure these numbers out and understand these 3 vocab terms is going to be crucial once we actually start talking about mitosis and meiosis. All right, everyone, thank you very much for listening to me, and I'll talk to you soon.